Hey guys, so I wanted to shoot this video for you as a quick follow-up to what we're talking about with DNA, specifically with its structure, and how that structure then leads to its function or the job that DNA actually performs. For a really long time, there was a huge debate about whether or not DNA was actually the molecule that was involved in heredity and passing along traits from one generation to another. The main thought there was that the molecule was just way too simple. It seemed kind of inert, which means it doesn't really like to react. It doesn't do much. It just kind of sat there. It didn't do anything interesting. And all the data suggested that proteins were the ones that really did the interesting chemistry and they did the really interesting things. Now, they were right about that. Proteins are the ones that do the really interesting stuff. DNA does just kind of sit there. But when you think about it, that's actually a really good thing, right? Because if DNA is just sitting there inert and it doesn't react with anything, it's able to protect information and pass it along from generation to generation. So, as soon as the structure was discovered in the 1950s by Watson and Crick, along with an incredible amount of help from our favorite scientist, Rosalind Franklin, they were able to discover structure, which immediately, when scientists find the structure of a molecule, they start thinking about, well, what does this molecule do? Because shape in biology is everything to do with this idea of function and how it works, right? So once the structure was there, things started to get unlocked really, really quickly. I want to re-emphasize in this video with you some important parts for us in BioTech 1 about the structure of DNA. There are some things that I'll leave out in terms of the entire story, but it's just because we don't need them or we're not going to use them in BioTech 1. We'll add detail at some point if you decide to go on later with BioTech, but for now, this is all we're really going to need to know. So this is a good summary of the things you should have gotten from the videos you've already watched. Now, the main thing is the monomer of nucleic acids. What are they? By the way, the Na in DNA stands for nucleic acids, right? It's just a type of molecule, really, really big, long molecule. The repeating units in DNA are called nucleotides, of which there are several components or pieces. The first is something called ribose, which is a type of sugar, and it's got 5 different carbons on it, and that's going to be really important for us later on in the class. So here you can see there's a little oxygen atom sitting right at the top, or the crown kind of the sugar, and then here's each corner here is going to represent a carbon, and then the 5th carbon is actually represented right on top there. I'm going to point these out now because they're something that we take advantage of later on in biotech by knowing the structure allows us to do some fancy tricks and some application, which is really what biotech is. But anyways, here's your 5 carbon sugar. 1, 2, 3, 4, and then there's the 5th carbon sitting right up there. And that's going to make up part of the backbone. The other portion of the backbone is going to be something called a phosphate group. And this is acidic. And really all an acid is something that donates or gets rid of these things called hydrogens, these H pluses. They go away into water. This is an important part to know here, that phosphate, when it doesn't have these H's, like whenever it's in water, it's going to get rid of those H's and it will become negatively charged. And this phosphate group being negatively charged allows us to do lots of other really cool tricks that we'll take advantage of here in BioTech 1. So that's an important thing to mention. The third component is really what determines the flavor of the nucleotide. And that's the nitrogen-containing base, the base group, or not an acid, right? The base. It determines the identity of a nucleotide, and there's 4 different types. There's a 5th in RNA that we'll discuss at some point, but for now, the one that I'm showing you up here is the example adenine, or what we oftentimes abbreviate as the letter A in the genetic code. Right? So there's adenine, and it makes these 3 parts, and now we have a nucleotide here of DNA, the phosphate group, the sugar, and then also the base. Notice guanine here. It has a very similar structure to adenine. When I say a similar structure, what I'm looking at specifically is obviously they're going to share the backbone components. Those are similar to all the nucleotides. But go ahead and look at the nitrogen bases. They each contain these 2-ring structures, which make them actually very similar to one another, right? There's another type called thymine. And you'll notice the slight difference here is that there's only this 1 ring that makes up thymine. And then the last base is cytosine, which also has the 1 ring, which makes it a lot like thymine. So these are our 4 different bases in DNA. Okay, the next thing to look at is there's these covalent bonds, and that gets into a little bit of chemistry. Really all it means is they share some electrons. If you don't know what that means, just think of a bond as magnets, like kind of they're sticking together, right? They're forming a unit with one another. Covalent bonds are going to form between a sugar group here and a phosphate group on the next nucleotide in the chain, right? So they're ultimately linked together, and the fancy name for that bond is called a phosphodiester bond. When they're joining together between these two groups, and that's shown right here in these arrows. That's how they connect together. When you get a bunch of phosphodiester bonds together, this bond between the phosphate and the sugar, you can get a long strand of nucleotides, and a long strand of nucleotides is what we call DNA, right? Deoxyribonucleic acid, really long strands. And then here what we're representing is the different bases. So this particular strand represents A, C, T, T, A, and G, right? And that's if we have one single strand connected by phosphodiester bonds with each other. We get really long molecules. Okay? Now, the next thing that I wanted to mention is that two strands are always going to join together in DNA. It's a double-stranded molecule. So on the other side, what we're going to have is a complementary sequence, right? We always are going to have certain nucleotides pairing with other nucleotides, and I think that's a rule that you guys all pretty much get, right? We've got A always pairing with T, and the main reason is A has this 2-ring structure and T has this 1-ring structure, and we always want 3 rings going across so that we can have a nice symmetrical, inert, non-reactive molecule. G is always going to pair with C for a very similar reason, right? G has these 2 rings, C has 1 ring, they form 3 going across to form these bases. And that's why we get these base pairing rules. Those bases are going to be joined together through hydrogen bonding. And that's an interaction between these bases that are going to hold them together. And that's how we get this double-stranded molecule sort of stuck together, right? It's through the hydrogen bonds interacting between these bases here. And I'll show you some other images that I really like of this idea. That's going to lead to this idea of complementarity, or the really fancy term for it is Chargaff's rule, from a guy named Chargaff that actually discovered it by looking at the relative percentages of nucleotides in organisms. Really, all that fancy terminology means is that A pairs with T and G pairs with C, right? So if we know the sequence on one strand, we can determine the sequence on the other. These things are also what we'd call antiparallel. So the only way that we can actually get DNA to base pair for this complementarity to happen is if they're parallel, meaning that we have them side by side, right? A line next to another line. But we give it the term anti-parallel because it's actually flipped upside down. So if one has a regular orientation, the other is going to be flipped upside down. And that's the only way that the bases can actually get at one another. So this diagram is showing you the anti-parallel nature of DNA. That one strand is going to go in this direction, and then the other is going to be parallel to it, but flipped upside down so that these bases can touch each other, and you can have hydrogen bonding. This allows for the function of DNA, which is really to be inert and not to react with much, right? So these bases are protected, they're facing each other, they're away from the outside world so they don't do any chemistry and they don't get changed. When we change bases in DNA, it's called mutations, and we'll talk about those a little bit later on in the course. Once you get all this chemistry working together, naturally, the lowest energy form of DNA is to twist up into this double helix. And let me move this real quick so you guys can see that. Right? That results in this twisting up structure. That's the really famous shape of DNA called the double helix. So you put all that together, and this is the shape that we get ultimately of DNA. Okay? The sequence of the A's, the C's, the T's, and the G's on one strand contains all the information that we're going to need in order to make something called a protein, which we know about now. It does all the interesting things, right? And when things go wrong with proteins, usually what we can go back and look at is maybe something that went wrong with coding in the DNA or a mutation. Now, this is just a little bit closer look and a little bit more detail of what's going on between bonding in nucleotides in DNA. So here, just as review, we've got our phosphate group, part of the backbone. We've got our 5-carbon sugar, part of the backbone, and then we've also got our nitrogen base, right? Now, what I'm going to do is progress through a series of images here that are going to show you how this all interacts, okay? Now, the next thing that we're looking at here is we're going to also have a nucleotide. There's the phosphate group, there's the sugar, and there's the base, right? And those bases are facing each other, and they're going to want to join together with hydrogen bonds. So that's what we're representing by these dots here, right? We've got these two places are where hydrogen bonds are going to form, and now these nucleotides are stuck together, the A and the T. The next thing that we have is we're going to add another nucleotide. So notice what I did here is I just added another nucleotide, and we formed a phosphodiester bond. here between this sugar and this phosphate group. And that's forming the backbone, right, of the DNA, right? And then the rungs of the ladder are here. There's G, it's going to pair with a C, and then they're going to form hydrogen bonds in between these, three areas here, right? And now what we have is an antiparallel strand of DNA, A binding with T, G with C. Okay, that's like the basic idea. There's one last image that I really like from Molecular Biology of the Cell, a different textbook that I wanted to share with you guys that really show the building blocks, DNA building blocks. Think of them like Lego pieces. They're always repeated and they get you a certain shape, right? The first is this sugar and this phosphate together, and they're representing it up here with this orange sort of triangle, and then the phosphate is like this yellow sort of sphere. These two things linked together will combine with a base, and then you get like this kind of this shape, right? Which part of that is the backbone, and then the other is the base of the strand. Now, we can start linking these things together, right? Where on one end we have a phosphate group, followed by the sugar. Then there's another phosphate group, which this belongs to C here, and the sugar. That's going to go ahead and connect to another phosphate with the sugar, that is A's, right? You guys see how these kind of like link together. It's a nice image that I really like here of how they actually link and form together. These bases then are sticking out, ready to join with some other bases, and then that's what you're seeing right over here is the hydrogen bonding between these bases. Perhaps a pattern that you're noticing is that between G and C, they form 3 hydrogen bonds. Between A and T, they form 2. And we'll talk about how that actually has some applications for us in biotech a little later on also. But notice here, right, that we've got on this direction, there's something called directionality, which we're going to talk about relatively soon as well. Where here, we've got our phosphate group on the bottom, and we've got our sugar down here. The antiparallel nature of the molecule requires that now the other molecule must have its phosphate group up top here, with the sugar ending down here on the bottom. You notice how these are like mirror images of each other, right? They're anti-parallel. That's going to be a big application for us later on that I wanted to point out, and something that I think is particularly useful with this image. Okay, so hopefully that helps clear some things up. Those are the important features of DNA structure for now. We're going to manipulate and take advantage of all these structures by doing a little bit of chemistry here in class. You started by pulling some DNA out of cells this week. Now next, we need to take advantage of some of this chemistry so we can do some steps downstream.